I'd like to welcome you all to the Center for Complex System Studies here in Utrecht, and to this masterclass on neurobiological networks. And I'm like to introduce you all your speaker for the next 3 days. That is Daniele Avitabile. He is a professor at the Vrije Universiteit in Amsterdam and a member of the Amsterdam Center for Dynamics and Computation. He did his PhD in 2008 at the University of Surrey in the United Kingdom, and then he spent time at various institutes, I think Nottingham and Bristol, maybe in the UK. Came to the Netherlands in 2019, joined the university there. His research is very broad and covers many aspects of applications in neuroscience, but it includes modeling, it includes analysis, it includes computation. He has expertise in numerical bifurcation analysis, in spatio-temporal patterns, in nonlinear PDEs, multi-scale techniques, and numerical methods. So he's really an expert in this area, and it's a great pleasure to have him here to give this course. So,
thank you very much, Daniel. It's a pleasure being here. Can I just have a quick idea of who am I talking to? Because I think this has been my biggest challenge for the past 2 or 3 weeks, to try and understand who's the audience to whom I'm talking. And to a great extent, the lectures will be actually dependent also on what you want to find in them. So by show of hands, how many of you have ever modeled a neuron or know? Yeah, so you perhaps have heard of Hodgkin-Huxley modeling neurons. I'm not expecting anything. I'm just trying. How many of you have done or enjoy doing numerical analysis as improving convergence of a scheme? I expect nobody. 2. Okay. And how many are into sort of nonlinear analysis? Like, do you even know what it is?
Okay. For me, like nonlinear is more like for FEM, it's like a materials and not electrical.
Okay.
In that sense.
And ultimately, how many of you are here to expect some form of hands-on approach, or more theoretical? If you were to say hands-on versus theoretical, hands up for hands-on. There's a lot of, I think that may be all right. So, so what I want to do, let's see if this works now. Yeah. So what I want to do in lecture 1 is to give you a little bit of an overview on modeling neurons and cortical patterns. This is partly taken from a first lecture that we do in the Master Math course on mathematical neuroscience with my colleague Hilmeyer, and it runs every 2 years. But I'm going to give you a little overview of how we model neurons and how we have given up trying to find an all-encompassing model for neurons, and instead concentrate on selected models. And I'm going to try and give you an idea of how patterns in brains are formed, both experimentally, what we observe, and I will concentrate, at least for the first lecture, about 2 mechanisms for getting patterns out. So we're going to look into 2 types of models. One of them is called neural fields, or firing rate models. That's going to be the one we're going to stick with for the rest of the course. But today I will also show you some, I think, very exciting things that happen outside of neural fields, at least for today, that have to do with spiking neural networks. So this is something we're going to find out later. And the tutorial is basically about discovering or rediscovering the formation of pattern through Turing bifurcations, if you've ever heard this term before. If you haven't, you don't need to know about it, in neural fields. And then the other 2, we're going to see how it goes. So I do like being interrupted at any time, so please do so anytime you don't understand anything. So I thought, what will I do? I will just give you a 5 or 15, 20 minutes introduction about brain modeling for those who haven't seen it before. And so this is stuff that I fetched from the Brain Facts book here. So of course, that's the organ we're going to be trying to model, or part thereof. It's 1.5 kilograms in weight. It's about 1 liter, more or less, in volume. And it contains an enormously large amount of neurons. These are the cells that are inside the brain, and an even larger number of synapses that are connections between the neurons. We're going to see a couple of videos with that. And of course, you might know that it transmits information and generates many things between motions and emotions just by sending electrical signals across the brain. And it's actually a power-hungry beast. It consumes 20% of your body's energy. So I thought I would start by, how can you not start from that? You would start from telling you, showing you the first images of neurons that have ever been, that are known to humanity. So we are around the 1900s, end of 1800s, I would say. And Golgi has just developed staining techniques for cells. So at the time, things were black and white. And Ramon y Cajal looks at and learns this technique, it perfects it, and then it starts drawing what he sees. So this is a drawing of what Ramon y Cajal saw through the staining technique of a particular type of neuron. If you have not seen this before, I strongly encourage you to Google Ramon y Cajal. There are just mesmerizing images that come out of a Google search. And of course, today we have very different and more powerful ways to just image neurons. So, oops, that was meant to be a YouTube video. So what you're going to see here is images that are reconstructed from effectively, I would say, a section, more like a carottage. So they're just putting, if you want, a needle in through some parts of a brain. You'll see this later. And from it, you see that the first thing is that not all neurons are, there are different type of neurons, say. So this is neurons that come more at the surface of them, below the surface of the cortex. You see that all of them are little buttons that where the nucleus is located. Those are called the somas. You'll see more and more of them. And they are arranged in families that depend very strongly on where they are in space. So as I say, this is now not really a section, but more like a column, so to speak, underneath the cortex. And you see there are different types of neurons, not just densities, but different conformations of neurons. And as you may have heard before, not only do they come in different types neurons, but they're also located more anatomically in different parts of the brain. So of course you may have, well, not of course, but you might have heard about different lobes of the brain, the frontal lobe, the sulcar, the parietal lobe, the occipital lobe, and the temporal lobe. We're not going to deal with any anatomy. Don't worry about that, because I wouldn't be a good speaker for that. I think that the section we have just seen is something that, in my opinion, goes through this part. So near the corpus, from here, it just enters the corpus callosum, and just enters more or less around there. Now, what we do typically in the very first lecture of math, in the MathNeuro Master Math, is we show the following video, because actually it's very impressive. And what you're going to see there is an actual image of a small portion of neurons that live in the prefrontal cortex. And in it, you will see some form of coloring and connections that show links to and from the amygdala. This area there that is underneath, so the cortex is not really a cortical area. So I'm not sure that the audio, I'm not sure that the audio will play. We'll find out. I have to be patient. See if this works. And
everything you see here is real data. We're looking through microscopes at neural tissue going down through an area of the brain called the medial prefrontal cortex. We can use these microscopic images to reconstruct the three-dimensional layout of neurons and really see the intricate and intertwining patterns that they make.
So effectively, you're seeing the two types of colors here that I mentioned earlier on.
And are responsible for everything. Control your muscles, how your sheets, to how you feel about your family. Understanding these patterns is a massive ongoing project in neuroscientists everywhere. It could lead to breakthroughs for everything: consciousness, depressive disorders, and schizophrenia. In this example, we injected purple dye into a far distant brain region called the amygdala, which is largely where our emotions, feelings,
So,
fear,
what you see in purple is stuff coming in from the amygdala.
or connections coming from the amygdala into this area. We've also used advances in genetic engineering to select inside this region that connects to the amygdala and dive.
In green, this is the connection that are coming locally to, that are going to the amygdala.
That connect to the amygdala and connections coming back in from the amygdala. The locations, patterns, and neural connections called synapses.
The synapses, the connections are these things that are in
To help us
science, I would say.
These neural networks. We develop computational techniques.
And we stop the video here also because there is a tedious people. Let me try to mute myself. So the basically take-home message that you should get out of this, if you've never seen this before, is that, well, this was in the second part of the video, the neurons communicate through spikes of activity. This is something that perhaps I should have made clearer before, but it's not there in the video. Of course, you see the neurons are wired to each other. There are different types of connections that go in a neuron from far away or locally as well, but there are also connections that go far away out of a neuron, and that there is an enormous amount of connections between them. Those are the cyan spots that you've seen earlier on. Those are what we call synapses. And importantly, perhaps, is that these connections are highly non-local. So in terms of sizes, yeah, you were looking at sizes of perhaps 0.1 millimeters in size, and the connections were just moving away centimeters. So this is literally a different range of scales that you're looking at. And as you will see in the next slides, we also need different temporal scales to resolve what neurons do in principle. And perhaps the most compelling message, in my opinion, of that video is that we really need a collaboration within multiple disciplines to make sense of that. So we need biologists, we need engineers, we need mathematicians and physicists. So what I promise I'll do at the beginning of today is just give you a very, very quick crash course towards what type of neuronal models there exist at the moment. And we have to make a choice, as I stated in this course, but also in science, about what scale we depict, because I should tell you straight away that the hope of modeling a brain, starting even from cells up to all organs, is just, we're not there yet as a community. And this may seem kind of a bad message. I consider this to being a fantastically exciting message. It turns out that at the moment, if you try, we have different models of neurons as cells, single cell, we're going to discuss that straight away, but bridging this up to region is turning out to be effectively not just mathematically challenging, but also the type of models that we get at the end, they are also not manageable. So in the rare cases where we're gonna take, let's say, a neuron with all bells and whistles and couple millions of them and trying to get a mean field equation, even the mean field equations seem to be intractable. So the bottom line is that I don't think we are there now having a Navier-Stokes equation equivalent for the brain. And mostly of what we do is we try to look at these problems from different sides, from seeing different sides, so to speak. So where to start? If you want to model a neuron, I think this is a fantastic the picture that is in Izikiewicz's book. There is a set of references at the end of my slides. It shows what typically happens between two neurons. Immediately you can see that the neurons have the soma there, that is where the nucleus is kept. So if you think of a cell as being the classical rounded shape, you would just only look at the soma there. But a neuron has this kind of long branches, and there is a difference in branches between branches that convey output in a neuron. Those are called dendrites. We're not going to need this terminology, but just for you to be familiar. And they are far away, apical dendrites, so there are some dendrites around the soma. Typically, they're just the wiggly curves. That's actually, you can keep them, and they're conventionally the ones that convey input in a neuron. And then there is other wires, notably the axonal wires, that actually convey output. So in here, you're basically, before we saw both axons and dendrites, I think. Well, we saw axons mostly, I'd say. And they communicate through, so this is now an axon that is branching towards a faraway, potentially a faraway neuron. And it connects on the apical, sorry, on the basal dendrites of another neuron. And it does so through, let's say, a synapse. We're not going to get into what a synapse is. Just think of it as a connection in between them. And another nice thing about the slide is that you start seeing effectively what are the units, both in space and time, for neurons and spike. So this is, roughly speaking, 0.1 millimeter. The voltages that are measurable on the soma, on the cell surface, as well as on the axons, are of the order of -60 millivolts to 35 millivolts. And a spike really takes milliseconds. So this is a very fast time scale. So the first thing I want you to show is how you can try to model using a Hodgkin, the Hodgkin active formalism, neurons. And this is one of the great success stories, I would say, in applied mathematics. It was worth a Nobel Prize at some point to Hodgkin and Huxley. And the idea is actually, once you see it, quite simple. You can model a cellular membrane as being an enclosure that is made up of fat, so to speak. And this is a typically, well, it's a weakly permeable boundary between an inside of the cell and the outside of the cell. And the main idea is that this boundary, when all channels and all porosities are closed, it serves at rest to separate ions on either side of the cell. So this is a depiction of just what happens to potassium channels. So that's the potassium channels there. This is the inside of the cell. That's the outside of the cell. And for the time being, imagine that there is no channel there. At rest, there will be a separation of charges between the in and the out, and that is typically modeled as a capacitor. So the capacitor is just I would say, an electric gadget that is capable of keeping to separate charges on either side. So this would be one side of the capacitor, that would be the other side of the capacitor. Vm in these slides is the voltage membrane. And so there is a constitutive relation for a capacitor current that says if there is a current, then that will be proportional to the derivative in time of the voltage membrane times the constant Cm. Now, what happens when physiological conditions want you to do so is that channels open. And when the channels open, they let some ions in. And therefore, the way to model this thing, at least at the first level, is just to put a resistance there and to consider this as just a resistive current, So this is what happens there. It's Ohm's law here is written with not with resistance multiplying I, but with conductance multiplying V. That's typically a convention that is used in this field. You'll see that at rest, there will be a voltage that is a reference voltage. It's called EK there. And so effectively the simplest cellular model you can think of is really one that views the cell in that way. There is an outside and there is an inside. There is a difference of potential Vm. This stuff may change in time, Vm of t, because Kirchhoff's law applies. So there is a balance between the capacitance current and the current that is induced by a channel opening. So at the very first stage, this is not a good model, but you can, it's a good initial model. You immediately can write down a differential equation that evolves the voltage of the membrane in time. So in this picture, C is a constant that is given to you, G is a constant that is given, and E is a constant that is given. So this is nothing but the equation x dot equals minus x in time, well, risk plus a constant. So it's a linear ordinary differential equation that evolves the voltage. How is that? I mean, I need to have some feedback. How is this going? Okay. So if Hodgkin and Huxley stopped at that, they wouldn't have won a Nobel Prize, because the only thing that can happen to a linear system in this regime is that they will respond to an external input. And in particular, they wouldn't be able to just do a spike of this type. For this to happen, you need some form of nonlinearity in the model. And how do they decide to model the nonlinearity? Is to say, well, first off, the picture that I gave you before, it's obviously only the picture in which I think I put there a potassium channel. There will be a variety of channels. I think in the model that they pick, they consider sodium, they consider potassium, and then there is an L current, which is just the leak current. So let's say the top part of the equation is just a repetition of what you've seen before, in which you add more and more currents, because now you have more channels in there. But what they did, they had the intuition, is that now, for some linearities to happen, you need to want your channels to behave non-linearly. So the idea is that, remember the gating that you've seen earlier on, the nice little channels that let the potassium through. I told you, initially we depicted them as they were constantly on or constantly off. But it turns out that they are on or off depending on voltage. So the typical way in which this is done is to say, well, I'm now concentrating on the alpha current, alpha being, let's say, sodium or potassium. And the only thing that I've done with respect to the past slide is I've stated now the conductance is mediated by two variables, M and H. They are called gating variables. They describe whether that you can think of them as being numbers that depend, well, scalars that depend on time, and they are between 0 and 1. So they are 0 when the channel is activated, and this one is, this one is 1 when the channel is activated. This one is 1 when the channel is deactivated. There is a little bit of a dance, and I don't want to get into that. But these variables themselves have evolution equations. So there are now nonlinear functions that depend on the voltage. So their characteristic opening times depend on the voltage in complicated nonlinear fashions, and also their rest value, let's call the asymptotic value as they're called, they also depend on nonlinearly on V. And now the best way that I have, I'm embarrassed at how fast I'm going. The best way that I have to offer to you, if you've never seen a Hodgkin-Huxley model before, this will be the end of it as far as we're concerned, is to think of them as being a nonlinear evolution equation for V, in which the currents are many, depending on which channel you want. And for each channel, you also have to specify opening and closing of the gates. And then you stack up this set of ordinary nonlinear differential equations, and you move them forward. And they can, in fact, exhibit quite a bit of the scenario that are seen in cells, both in vivo and in vitro. So this is a typical thing that a neuron will do. It will emit a spike. That's really without this, a neuron would not be a neuron, I would say. But it can also have a variety of rhythms. So this one is now what happens if you have periodic oscillations. The periodicity can really be nonlinear, or it can vary. And there is an enormous amount of rich variety of patterns that you can study just by effectively introducing the right channels with the right model. And this is actually a phenomenal success story, because, as I said earlier on, it just is able to characterize many phenomena that we see at single neuronal level. Any questions so far? I've not been interrupted, which I don't know how to read. Yes.
The I alpha on the left.
This?
Yeah, what was that again?
So the way to read it is that this can be a current, I for current. It can be a current of calcium, sodium, or potassium. There is no calcium in this. So this is an index that can be either K plus or Na plus. And then in each of them, there is constant, G calcium, G potassium, and there is an E calcium, E potassium. This is another constant. Then there are two variables, M and A, to some power A and B. And these are now the gating variables for the channel alpha.
Okay. Thanks.
So we're at single cell, and you have now seen the Hodgkin-Huxley neuron, and the evolving variables for Hodgkin active neurons are basically voltages and channels. I think this is the bit that I conveyed. So what evolves in time are voltages and these gating variables. In addition to an enormous variety of behavior being achieved by an increasing variety of channels, this is really a trend that the mathematical neuroscience community, and I would say the physicists working in the neuroscience community have taken to explain every tiny little behavior of a neuron. There has been another trend as well, that is the one of trying to simplify the model of a neuron in preparation for forming a network of neurons, and also because they're mathematically interesting on their own. One variety, we're not going to go through, I'm not going to show examples of all of them, but this math is important. One variety is just obtained by simplifying channels. So it turns out that one can get, so I should have stated this, that the Hodgkin-Axley neuron, even the simplest one that you've seen, is hardly tractable. So if you're one of these scientists that like to prove theorems, this is going to be hard, except that you prove existence and perhaps well-posedness of the problem, but there is nothing else you can do. So they're often, already with a simple Hodgkin-Axley neuron, you have to resort to effectively numerical simulation for that. But you can try to abstract out features that neuronal model have so as to be able to find founding principles. And that's the route we're going to be taking, but for the kind of neuronal networks. So if you try to simplify channels by simplifying the type of currents that you get, and perhaps to simplify the type of voltage gating that you get, you're going to get simpler model. You might have heard the name Maurice Lecard. If you haven't, at least you can put it in a map when it will happen to you. And you can even go a further simplification down and just retain voltage on its own and have an additional variable that mimics a channel, but it is not quite a channel, to get a so-called Fitsu-Nagumo model. And by the time you arrive at Fitsu-Nagumo, you really have the perfect mathematics, the most abstract mathematical description and simpler mathematical description that can give you these spikes as well as oscillations, so to speak. You can decide to take another route, that is to say, anyway, I've seen a spike, and a spike has this kind of behavior that it goes up and then it stays there for very short and then it peaks down. That typically happens because there is a nice syncing or rather dancing of opening and closing channels. So whenever a voltage spike goes up, there is quite a lot that is happening at the variables m and n that are the gating variables. But sometimes that can be an overkill for your application. So sometimes what you decide to do is to have a different type of model, they're called integrate and fine models, in which you do not describe a spike, in the sense you don't follow voltage going up and then down in a spike. You just decide that when an event occurs, you mark a spike with a dot. And these are tremendously successful neurons, some would say, and you will see these in the second part of my talk today. So at the very end of today, you're going to give you a hopefully an interesting way in which these type of neurons can make patterns. But for most, okay, and of course, especially if you are both mathematicians and the physicists community have investigated all of these with the addition of noise. So they can turn them into stochastic differential equations and study that. But for most part of this tutorial, we are going to stick with rate models. Rate models are models in which voltage is not even it's not even an item. The only thing that these rate models will try to do is they will try to describe firing rates. So they're assuming that neurons are there on or off, and they will try to describe rates of spiking. And there is an enormous amount of things you can do with that. And I'll get back to why these are decent models and what are their pitfalls throughout the talk. These 3 or 4 days, in fact. It's 3 days, sorry. But I think I have to, if there is one model that you have to know a little bit how it is derived, it's this model rather than the Hodgkin-Axley model, mostly for this course. So in the mathematical neuroscience course, what we do is through the course of, I think, 5 hours of lectures, we explain to students what compromises you have to make to believe a firing rate model. So we are just making no mystery of the fact that these are gross simplifications, and some of these assumptions are really not viable, they're not believable, so to speak. To the extent that we have stopped even considering them being bottom-up models, we consider them being top-bottom models, in which are phenomenological models as opposed to do the averaging. But so this is how the story basically goes. So I'll try, there are better ways to explain it, or quicker ways to explain, longer ways to explain it. But let's see how this goes. So the idea is that if you observe a neuron and you're now depicting it with its dendrites there and with its axon there, and you're now considering to impart an external stimulus that is for now constant, imagine I is just an external current and it's kept constant at all times. Depending on if your I is large enough, you're going to see it spiking at a certain rate with some sort of rates. And this, I think it's fair to say that's what we see in experiments too. Then you increase the I that you get there, and you very often see an increase in firing rates. And this immediately points to some form of relationship of R, which of course in here I'm making conveniently nonlinear because I know where I'm headed, but this is also fair. It happens sometimes, often it happens like that. And there is a relationship, which I'm going to call a firing rate function, between the intensity of the incoming stimulus and the rate that you observed in a neuron. I would say, of course, this is made up, but this is not a bad approximation. And it's actually quite nice that you can do that with however sophisticated your neuronal model is. So if you are non-compromising at this stage, and for you a neuron is the Hodgkin and Huxley neuron, you can, using bifurcation analysis, you can trace these curves by following in parameter space what happens. And perhaps the shapes will be different there, but that relation is there. Yes.
So here I is just intensity, not necessarily currents.
It's basically this is only a cartoon. I'm thinking of a scaling parameter for the intensity. But in a moment, you'll see it going. So what is happening basically is to say what typically happens is that current is proportional. It happens at a synaptic point. And I promise I would not model synapses in this course. But now that you have seen gating variables, you're halfway through to modeling synapses. So synapses are not strictly speaking channels, but you can model their being open or closed through some gating variables. So we saw earlier on, we called them M or H, I think, and now there is this variable S that tells you you're open or you're closed, how much you're open and how much you're closed. And so typically the thing to retain in memory is not I there, but rather S being the synaptic variable, being open or closed. So you can even imagine that there is like an external current existing there, whatever is the time dependence of it, but if my synapse is shut, it will not have any effect. And so you see immediately that in this new kind of form of models, I'm not even trying to model voltages of a neuron, right? Before I was really resolving what was happening here up and down in voltages, and now I'm just considering the rate at which a neuron spikes and a synapse. Okay, so now the burden has shifted from modeling voltages to modeling synapses. And so I'm going to have to tell you what is an evolution equation for a synapse. And the way these models are conceived initially is quite simple. They would say, well, let's assume that a synapse actually responds to an incoming current. So now you should imagine that I am looking at current getting into the axon, and this current has this form. It is a spike that is being received from another neuron, and this spike is impulsive. And just because I want to keep things as simple as I want, I will literally draw a synaptic variable of this type. Now, this is a harsh. It's a bit too harsh, perhaps, towards modelers in that respect. What I want to stress is that the synapse, it's not even open. It cannot do anything before a spike arrives. And when the spike arrives, in this particular model, there is an exponential decay. There is an instantaneous kind of jump in synaptic variable that then gets dissipate, that decay with an exponential rate, which formally justifies an evolution equation of this type. Of course, mathematically, one has to be cautious because this function is not differentiable. And so this is only a formal, and perhaps I have to reinterpret these solutions in a weaker sense, but who cares? There is basically an evolution equation for the synapses as a function of time, and there is this is what happens to an impulse there. Now, if you, I should say immediately, if you're bothered by the discontinuity as I am, you could also decide to model this in a different trend. You could decide to just model it with an upward phase and a downward phase, and it being continuous. And what you would do at that point is you would no longer have this simple linear operator on S, you would have a second order operator on S. So then there is possibility to do things more realistically. But let's stick with that for the time being. So now you are in a situation where you no longer have a single spike, you have a train of spikes, tau 1, tau 2, tau 3. And what happens to your evolution? This is a linear equation, so you're forcing changes with time. And each time you receive a new spike, you are reset up a certain amount, and that's now your synaptic variable, S of t. How is this going? Too fast? So far, I've told you things that are almost, that are effectively, I'm modeling the synapse, but I've not really made any approximation except modeling approximation. And now here is the bit that begins to be a little bit dodgy about rate equations. So the picture you have seen is a picture whereby the time it takes to a synapse to relax down is comparable with the time that I've drawn two consecutive incoming spikes. So what I'm trying to state here is that the synapse that I've chosen, in here I put like a scale of order 1 in terms of time scales, is more or less of the order of magnitude of that separates two incoming spikes. But I could, just out of mathematical convenience and nothing more than that, I could dream up a synapse whose time scale is so slow that it takes a long time to react when it gets n or m spike trains, one after the other. In that regime, what I could do is I could introduce an average variable. This is now the average over a time interval, capital T. I could make my synapses slow. And following this approximation, that is really the hard thing to believe, so to speak, one arrives at a very nice evolution equation in which there is no longer a Dirac delta function there. There is just a rate variable. This is now the rate at which my spikes are incoming. So I don't think I'm explaining to you what happens and why. I don't think I can do that given the time that I have today. But that's the gist of it. And you have to buy this slow synapse approximation to be able to do that. But now perhaps you see, but perhaps you see where this is now headed. A moment ago, I told you that the rate is a function of the synaptic variables. So I can now replace R of t by that firing rate function f of u, and I'm going to get a closed form equation for you. You'll see this happening in a moment.
So is S a voltage here, or is this?
Nope. S is really like a gating variable. So you can, physically it has dimensions of conductances, so to speak. So it's basically, so this is also imprecise. So if you remember before, at some point I had a conductance multiplying voltage, the difference of voltages, and that's how we regulate an incoming current. What this is now a factor that multiplies the conductance, so that either increase conductance and therefore increase currents, or decreases conductance and therefore decreases currents. So morally is a gating variable. It says, we call it a synaptic variable. So I will just do this in a second, but effectively this system is not closed because I would want R to be a function of U. But at this level, in which I'm seeing just a single neuron, I'm modeling one neuron, right? And this is the external synapses the external rate of the stimulus, so to speak, this is now an equation that I could go and simulate because R is just an external model object. What happens if I have many neurons now? I have neuron J and neuron I. Now the synapse travels from neuron J to neuron I. I attach to this synapse a strength, a weight, Wij, just because I'm getting ready to strongly linked populations or weakly linked populations. My synapse is now the synapses of the I neuron. So I'm now looking at the synaptic variable of this neuron as it receives a spike from neuron J. And I can introduce the same approximation as above. So what I have now is I will replace this term here. Well, first I will replace SI by UI, because I'm now looking at the average rate of the neuron, of the synaptic variable. And I will replace this by the rate. I'm just following the same approximation there. And this is where things will now close. So first off, you start seeing that this looks a bit like a network. And again, I have this term, RJ, that in the past incarnation, in the past model, this was an externally driven stimulus, whereas now I have to make this rate come from neuron J, so to speak. But that I can do, because if you remember 2 slides ago, let me just go back to it. 2 slides ago, I introduced this function, f, that is the firing rate function. Oh no, I think I'm victim of my own keynote. A moment ago, we discussed the fact that there is a relationship between the rate that a neuron exerts and the synaptic variable through a function that is computable, that we call the firing rate function. And now I'm just literally plugging that into here. So now Rj will become a function of Uj. And this is how you obtain a network that is self-contained. I think if you miss a little bit the steps that took me there, first, I'm with you in the sense that it takes hours to go through this and it's a bit fast. But you can also take another approach, that is to say, okay, we're gonna accept these as models of a neural network for now, with big caveats. Why are these models so successful? Let me show you very quickly how people have used them to cook up, literally cook up neuronal models at different scales. So you now saw me describing a simulation, well, a model with a network of neurons, in which UI are now the synaptic variables for each of these neuronal networks. But of course, if I make some nice assumptions, namely, all the weights are constant and they scale with 1 upon n or the right power of n, I can send n to infinity and now write down a single differential equation that still sees my firing rate, that is no longer describing a neuronal network, but rather a neural mass, a single blob that corresponds to all of those neurons. It's sort of an average type version of it. And what I want you to retain in memory is that in going from here to here structurally, you still have this group type here that is inherited from the synaptic variable. Remember, we had this dot minus u is really coming from how I modeled the synapse. And this f is instead what type of neuron I have there, because the firing rate depends on the neural response that I get. And now I have a single ODE for a set of, for just that neuronal ensemble. But of course, once I'm at that stage, my appetite grows and I understand, oh, I can now model a situation in which I have multiple neuron populations. This is, I name them by alpha now, alpha going 1 through n, so this is no longer channels, this is the number of populations that you have. And what has happened to me is that I put an alpha symbol there. Now my weights will depend on I and j, but also on which population I am. So that's beta now for the population beta, how it's coupled to population alpha. And I have now a model that actually if I do the right recasting, is nothing but that, with a different numbering of the neurons and the unknowns. So I still have retained that thing. And I can play the n goes to infinity game here, and I'm gonna get a set of what I call neural masses model, in which now these connections that are called connectomes, you might have heard these words, tell you how A and B populations are linked. But who am I kidding? These equations are just nothing but those equations, right? It's just I'm relabeling stuff around. And there is an enormous amount of literature that just tries, and actually successfully so, match experimental data to these type of models, neural mass, neural connectomes and that, or the ones that we're going to be dealing with for the remainder of the course, which are called neural fields. And they are obtained by sending the number of populations to be a continuum instead. So now if I were to tell you, I could have walked in and tell you, oh, this is a model of a neural cortex. I have a cortex that is a continuum. This is now some form of activity variable at position x and time t. I have an integral that looks at what's the activity in other bits of the cortex, and how it's weighed from position y in the cortex to position x in the cortex. I could have done that, but I wanted to give you a little bit of a feel for how these models are coming about. Now, there are better and more sophisticated and more believable models that can take you from single neurons to cortical models. That first of all, there are better ways to introduce even rate models than the one I've done in such a short time. But also there are more sophisticated versions of neural fields in which, by making some assumptions at the level of single neurons, one can carry out the averaging procedure without doing the firing rate, without doing the synaptic slow assumption. And we're not going to get into that for the remainder of the neurons of this course, because all the mechanisms that we have to learn about pattern formation, and in my case, because that's what interests me, numerical simulations of the models, is incarnated in these models there. So I use these objects literally as toy models, even though they have been mapped, as you will see in a second, to real world data. Any comments or questions? Yes.
So going this way, when you're taking the number of little populations to infinity, is the idea that now you can basically just look at, instead of the connectome, you assume that there's just a distance function, like somehow.
So the way that these things are used is to say, when I write this model, yeah, I have to specify links, and there will be short range links as well as long range links. And now what experimentalists have done through tractography data on long distances, they can tell you the intensity of these links, which are not necessarily distance dependent, but they somehow depend, they count connections. You are in different lobes of your brain, and you say how many connections are emitting from here and incoming here, like the diodes are defined in this slide. So you have a sense for how large how weak these connections are, how strong actually these connections are.
Okay, but it's not a function of distance necessarily, it's
Uh,
a function of coordinates.
This is something that if you stick with me for tomorrow, we'll discuss more. In fact, even now, we'll talk about that. This is basically a modeling assumption that depends on which part of the cortex you're looking at. If you're describing regions, of the cortex, this is a fantastic illustration that I didn't make this morning, but I will keep it here. If you visualize this as being your cortex, a cortex slice, so to speak, if you're trying to model things at this scale, and if you're trying to model connections that run through the cortex, then those connections are distance dependent. That is typically what we see. But if you're trying to do is to model long range connection that exits the cortex here and perhaps re-enter the cortex here, these are the plot. Then over there, the distance, Euclidean distance on the manifold has to be replaced by, I don't know, Euclidean distance, perhaps. This distance from wires, and so that's why I am not committing to this being necessarily distance dependent objects. They can be even very highly non-local and non-homogeneous. Any more questions? Yes.
I have a question for my understanding, like a high current, high rate, and a low current and low rate.
Yes.
Is assumption of the best.
It's something that you see in, if you can do it neurons in experiments in which you just literally grow a neuron in vitro, and then you can inject a current in it and measure what's the voltage that you observe. This is how actually Hodgkin actually won the Nobel Prize. Not only did the modeling, but they also did the experiment on the giant axon screen. And so you can literally observe that if you put an impinging current that is very small intensity, the neuron will not really start firing. It perhaps will just displace the voltage a little bit. And then you increase it and you don't see much. You increase it, you don't see much. And at some point you increase it past the point in which you see this neuron starts firing at a given rate. And the higher, well, but not really the higher. At some point there is a critical value past which you keep increasing the intensity of the stimulus, and you see more and more and more. So you literally, you see it, and then you model it in that way. But I guess what you're telling me allows me to say, this firing rate has to be picked. It has to be fit from data, let's say, typically taken to be a signal. So I thought some of you perhaps were interested in doing hardcore probability theory. Perhaps I thought some of you are statisticians or you like measure theories. And if you're not, don't feel guilty for that. But if you are, that's the slide for you. So what happens if you decide not to be compromising in any way? That's possible. Well, it's possible to some extent. So I'm giving you a glimpse here about what happens if you don't take this route of the synapses that are slow. You take your Hodgkin active neuron that has got many variables. That U here means all the stack of variables that change with time. I'm putting a subscript T because probabilists like that. And what I'm doing is now I'm pairing neurons together through functions that depend on the variables J and I. And immediately I put a scaling 1 upon n there. So the weights are all 1 upon n there. So what you can show is that if you add noise in the form of some form of vignette process here to this network, you now get a large dimensional set of stochastic differential equations. And what people have been trying to do, certainly in a branch of mathematical neuroscience that is really more attached to I would say, probability theory, is to investigate what happens as n goes to infinity, number of neurons goes to infinity, to these stochastic differential equations. And what they find, in the physics literature, this is called as a McKean-Vlasov type formalism. Not that I know much about it, but that's basically what is being done in the literature. The mean field that you get, let's say in massively inverted code, the average activity U is a scalar variable, which is good, but it's now a stochastic variable, which has a probability law P ut. And the evolution equation for U depends on its own probability. So in the past incarnation, what you saw is that in here there was a term that was computable at time t. In here, if you want to make sense of this, you have to effectively compute the expectation at time t of U under its own law, under its own probability distribution. And of course, that's something that is determined by ut itself as you're progressing. So what I'm trying to state is that even the simplest type of mean field rigorous description, and this is a beautiful argument to go through, spits out stuff that is horribly complicated to start with. So this is now a rigorous limit, but we are still trying to develop, I think, schemes for running this numerically. Yeah? So this was my 2 cents for you know, it's understandable that people try to simplify this stuff because that's hard even once you take the limit. So there is an enormous amount of work to take this limit, but also once you have it, it's not necessarily a manageable object. If you want to know more about this stuff, we have invited in Amsterdam an expert in this area on McKean-Glasov type approaches, and he's going to give a one-day seminar on the 3rd of June in Amsterdam, just on this topic. So I've lost the slide. Now we're getting towards, I think, a little bit more the, I'm going to switch soon to the board for that. And I want to show you, we talk about models, a little bit about the patterns that we typically observe and we typically try to describe with this type of models. So what you're seeing here is one of possibly my favorite example of experiments in which some form of localized activity is formed. So the type of cortex that they're recording here is a very simple cortex. I mean, the model that I've just described to you can work for large cortices or for very localized cortex. And in this case, that's the bit of cortex in a Drosophila central brain that has to do with how the animal orients its head. So the idea here is that what in these experiments, these are marvelous experiments, what they do is they tether a Drosophila they put it in an arena. So they have a stick. At the end of the stick is a Drosophila, and it's in the pitch black dark. And what they do is they shine a blue light that they can vary. The arena in which the Drosophila is circular. So they move this blade of light, and they can of course see the animal wanting to move its head. And at the same time, they can record what happens in the area of the brain where the Drosophila is supposed to just encode head direction, which also is a circular and symmetric cortex. So the idea is that what used to be cortex like that, let's say, is now just a ring. Okay, this is literally anatomically a ring. You can think of it as being a cortex in the form of a ring. And what they do is they play different type of blades, play different type of stimuli, and they can record. So you should really imagine that there is a small circle around here. And as time passes, they see neurons being active here and not active there. Then the head direction changes, and now the activity of the neurons are concentrated from here to here. And they can do that, and they obviously plot it with time going horizontal and space, which is identified. So this is really a ring going vertically. And what you see is that what they observe is that the Drosophila, when it's in the pitch black dark, so there is no stimulus whatsoever played, the Drosophila has a sense for my head is in the middle. How does it do that? Well, it's not quite the middle, it's a little bit of this side, to speak. But there is a blue, I mean, this is effectively, the blue here is the firing rate that happens more or less somewhere in the cortex. Without the stimulus, the Drosophila has a way to sustain activity somewhere in the front. So it knows north. And how does it do that? It does do that because its neuron, the neurons that live here, they have a preferred direction. So this is something completely spontaneous. Without any stimulus, the neurons they fire a lot in what is considered to be north by the head. And they don't fire far away from the north, because if they start firing here, it means that the animal has changed its head direction. And then, of course, so this is a super endogenous thing. It has nothing to do with the stimulus. And then what they see is that when the stimulus changes, also the head direction changes. And by that, it's done some form of 360 degrees head that I don't really quite understand. I don't know if the mosquito can do that, or if perhaps it did that. I don't quite know that. The next, so the idea there is that this is now an experimental example of a cortex that can have a pattern of activity in the middle. The second experiment that is also very famous is the one that has to do with the visual cortex, V1. So this is an example. This could really be what happens in one of the experiments that I'll discuss later. So what is happening now is that I'm presenting to you a stimulus that is boring everywhere except somewhere in space. And I'm presenting here something that has some form of grading here. Now, with many inverted quotes, if I were to record what happens in V1, V1 is an area of the cortex that is here, it's about the size of a credit card. It is known that position in the visual space are mapped to locations in V1. So if now if I'm now visualizing what happens in your cortex, there will be some neurons doing nothing. They correspond to those encoding this position, and there will be some firing a lot. This is different from the previous experiment because it's an experiment in which I'm placing a stimulus somewhere, and then I'm exciting stimulus in an area of V1. This is what they do in the Chavan lab, where they can record this thing on primates. So what they do is they show this stimulus, they remove the stimulus, and see what happens to the core. They can image this in vivo. So what they see is that a localized stimulus generates a localized activity that stays even when you remove the stimulus. So this is another instance of the fact that the cortex itself has to be able to sustain on its own some form of localized activity. It's true that I brought the cortex there by the external stimulus, but then I removed it and it stayed there. So this is now an equilibrium that is sustainable by the cortex. And that they can do that with different orientations and see different angles there, different colors. And this sort of localized activity I would say, is one of probably the most popular experiments that mathematicians and physicists try to describe. It's, I would say, pervasive quite a bit in neuroscience. This is another experiment that has to do with an oculomotor task. So this is, again, I think, a primate that is trained to look at a particular area of the screen under a certain task. And again, this is a situation in which the horizontal axis has to do with motions somehow, in this case, the motions of the eyes. So this is also a sort of ring geometry in which a stimulus stays there. So this could be like a version that is more similar to the Drosophila example that you've seen earlier on. And finally, I often list this as, I mean, if there is one experimental paper that you want to read that is both modern and it offers a fantastic application for neural field models, I would recommend this one by Gonzalez Ramirez et al. From Mark Kramer's lab. What they're recording here is what happens, well, they're recording with 16 microelectrode arrays in patients that have epileptic seizures. So what they're doing there is that they're showing what happens in recordings just at the onset and at the offset of an epileptic seizure. And what they do, not only do they fetch the data, they also model, they actually show convincing evidence that there are waves of activity that are localized in space that span the cortex through the electrode arrays that they see, and they model that using neural field equations, like the ones that you've seen before. I think. So of course, you will see more patterns, I think, tomorrow, a little bit, coming from experiments. But basically what I have to do now is to first give you a primer for how neural field models generate this type of patterns. So I'm going to say, in all of the examples that you've seen in the experiments, it is feasible, or at least we're going to take neural fields as models of the cortex. And I want to showcase to you how these models are able to generate spontaneously patterns or not. And I thought this might be a nice activity to do. I'll do it on the board. The tutorial that you'll do later on today is really geared towards make you reproduce the calculations that I do on the board, and also doing some numerical experiments in which you see these patterns appearing. And then if I have time today, I'll show you alternative and very exciting ways in which you change the model. So instead of doing a neural field, you now take one of these spiking models instead. And then the mechanism for generating neurons is completely different. Do we need a break or can I move on? What do you normally do? People go on for 2 hours and we see people dropping off in the midst. What do you feel like doing? I need 20 minutes on the 30 minutes on the board and perhaps 20 minutes of my ego showing slides on a cool projects that I have. You want to take a break or should we move on? Just be open about it. Break? Small break. We reconvene at 1:15. Yeah?
Yeah.
Yes. So can I just quickly ask, how many of you have heard the term Turing bifurcation? Just hearing the term. Okay. No, okay, not many. So I'll just put a slide there that is, I'll get back to it, so there is no problem in, there is no need to do it there. So what is one way in which nonlinear dynamicists describe the formation of pattern? You need two ingredients from the wow. This is it's oversimplifying, but certainly you need to understand this, you need two gadgets. One gadget is a system that I have a handle on through a parameter. This could be the intensity of the impinging input or just one scalar variable that you can operate and change. And then what you need is some form of nonlinear medium that initially has a completely flat state. And then by changing the parameter, it starts developing steady states that have a spatial profile. So I think the thing I would recommend is sticking with this. I'll draw it in a second on the board. But the idea is that you have a parameter here, that's our handle. That is, I mean, I'm being vague on purpose here. You change the parameter, and for some changes, nothing happens to the zero state, it remains flat. And then this zero state continues to exist, but it becomes unstable. And if this term means nothing to you, we'll find out a little bit about it now, but it basically means that the system can exhibit this state, except it can't, because any tiny perturbation will take you away from it. That's the notion of, that's why I put it dashed. But nearby, there is some new states that have some form of patterns to which you can get attracted. And that's basically the mechanism for a Turing bifurcation. It happens in two flavors, supercritical or subcritical. We'll discuss that later. And so what I want to do is I want to give you a primer as to how our systems can give us a Turing bifurcation. I'll do that without being rigorous, because that would turn it into a horribly boring lecture. And I'll just instead, I will write true things, not worrying about proving theorems, because otherwise, as I said, this becomes a bit too overwhelming. But it can be done. So what's the main idea? We have a neural field. We have some synaptic weights, which for the time being, I'm going to keep very general. So I'm not making any assumptions on distance dependence or anything of that sort. And I need to put in a parameter. I'll put it here. P is going to be my parameter. And perhaps there is an external input, I of x. Now, the idea of those diagrams is that they put P on the horizontal axis and something that I'm not going to bother with on the vertical axis that has to do with a measure of an equilibrium. I'll populate this in a second. So the starting point, and also let me start making things easy. I'm going to take, do I need that? No, not yet. Let me take a cortex being generic for now. So what you see there is a map of equilibria. So what I'm trying to do here is, as P changes, I want to describe how equilibria changes. So I have to start by defining an equilibrium. Well, an equilibrium is a special solution to this problem, which is, I call it using a star, which is constant in time. That's what it means to be a stationary state. Are you okay with this or not? So now I'm going to have to search for special solutions of the problem above in which time is not there. And what I'm trying to do here is I'm trying to plot, I wish I could put u star here, but that is a function of x. So I need some form of norm of that. Let's say the maximum of that u star. So what will I do then? I will just start by writing down the steady state equation for that. So what I now get is I have to, if I want to seek for a steady state, I'm going to have to solve this steady state problem. I need to find a function u of x, u star of x. This function need not be flat. It can be also profiled. That satisfies this balance equation. And this is typically a non-trivial undertaking because the function u of x that you want is also appearing inside that nonlinearity there. The function f, I should have stated this early on, you can think of f of being a bounded function of this type, but literally it's not going to be very important for us. It's just a nonlinear function. And the thing that I wanted to appreciate straight away is that if I move from p to p tilde, now I made the parameter appear here, but I could have made it appear here. I could have made it here in front of p. This is just a floating parameter. This equation changes. Think p equals 0 or p equals 22. The function that I'm looking at is a different function. I would have to expect a different u star. And therefore, I'm going to traverse different points on this curve by just finding different solutions. Any questions on this? Because if this is not clear, you're going to get into trouble soon. Yes.
Do you have an example of what the dependence on P would be like a gain on a signaling or something?
In the tutorial that I'll make, I'll put P here. That's basically the gain, exactly. But it's not infrequent to take I to be constant in space and equal to P. That can be a constant. This is now the level that I'm now of current of external stimulus that I'm giving to the entire network, and it's constant, and its level can be P. But actually, it can also be the midpoint, the shift of the firing rate. That's why my instinct was to put it here, because the most famous example is that. More questions? Solving that problem is typically highly non-trivial, and we're gonna get to it tomorrow, I think. But for the time being, I'm asking you, suppose that I have a mathematical device that can produce U star for a given P star. Suppose you have it. What we have to do is to now discuss under which condition these variations of P determine my pattern to be unstable. So on the board and also on over there, you see that there is something critical that happens at some point as I change P, and that P changes in such a way that small perturbations to U star get amplified. So now I'm getting ready to say, well, what will I now do? I'm going to look at stability. And what is the stability problem? The stability problem says start at time t0 with the true solution that you just found up there, plus some small perturbations. So I'm going to call these perturbations u tilde of x at time 0. So this is my perturbation And all I'm doing here is I'm enforcing the fact that I'm specifying a perturbation at time t equals 0. And the idea is that this can be smaller. Now, to make sense of what it means small, we would have to introduce some functional analytical concept, which I'm not prepared to do. And so it's also a distraction. Think that these terms are small. You are just having an imperfection, some sort of experimental imperfection, or even your numerics causes this imperfection. You're trying to simulate a system in this region of parameters, and because of noise, you make some, there is a mid-fit between the true steady state and some perturbations. You get stability if you tilde x as a function of time go to 0 as t goes to infinity. So this was an initial perturbation that you make, and now you're interested in the response of your system when, after you bring it out of the equilibrium, what is going to happen? Will this term go to 0 as t goes to infinity, in which case I go back to the equilibrium, or will it blow up over time? I'm conscious that if you've never seen Turing bifurcation, this might be a big step. So can I just do a quick round of, do you understand this concept? Have I made it clear? So the way I want to picture with a film, perhaps, is that I have a steady state, u star of x, and at time t equals 0, in a color, hopefully, I just perturb this, and at each point over there, I have a perturbation u tilde x at time t. And what I'm investigating after a time t0, I do that, what happens to u tilde? Will it tend to 0, which means I recover my steady state, or will it not? And that's the idea of a stability problem. Yes.
Just like, but the stability could be have like a multiple status. One is. So
what your intuition is correct. Now I'm sorry that I have to switch on this side of the board, but in here you see that there is this here, there is one steady state, there's one u star, but at some point there will be more than one u star, in fact 2 in this case, yeah? But the analysis that we're doing, the bit that we're investigating is only when I perturb around one of them. And what I will observe is that perhaps I go somewhere else, that's where I go into the other state, if I am unstable. But if I'm stable, I go back to it. Yeah. Any more questions? So what should we do to just look at these states? Well, a typical thing that people do in linear stability is to plug these ansatz into the top equation, the evolution equation, and disregard nonlinearities. In other words, what you do is just recall that f is a linear function, a nonlinear function, and so you can use, to a first approximation, Taylor's theorem, and disregard higher order terms. So this is now what happens if you have a nonlinear function, and you're linearizing it around u. Remember, f is a real valued function, so it's real valued of real variables, and you're omitting this term. So what you do is you plug in the following ansatz. You postulate uxt to be u star of x plus u tilde xt. You plug this thing into the top equation. You have f of u plus u tilde there, and then you use this formula obliterating the higher order terms. And that's called linear stability because it gives rise to a linear system, as you will see in a second. And it's typically valid, this approximation is valid provided your perturbation stays small. So what do you get if you put this one there? You're going to get the following. So in here, I should have differentiated in time u star plus u tilde, but u star does not depend on time. So let me write it like that. Now I have minus u star of x plus u tilde xt. Then you're going to have plus the integral of wxy F U star of y. In dy, this is what happens when I'm now plugging. I am just retaining this term there. So the first term gives rise to that. There is going to be a second term, which I better write down here because it's hairy. With a minus sign. F prime at U star y U tilde yt. dy, I stop here because I'm truncating up to this term, so I'm not taking the second order term. And then there is I of x, which has always existed in my equation, which I put on the top. So all that's happened is that I'm now seeking for special solutions of this type under the assumption that I stop at first order with the Taylor expansion. Yes?
Is there a reason why we got rid of the parameter p?
Laziness. Yes. Okay, am I allowed to not? Yeah, that's a good point. I should have. But perhaps one thing to retain is that I'm not even trying to perturb P. So over there, I stopped somewhere. Yeah, you're really good point. So this, I stopped somewhere there and I'm linearizing around that point. Thank you. So I'm halfway through because remember, U star is a steady state. And so what it means is that it balances this term there. That's what you typically expect in a nonlinear equation, in a nonlinear setup. And now you're left with the following linearized problem. U tilde xt equals minus U tilde xt plus an integral of w x y f prime U star y U tilde y t in dy. And this may look horrible, but actually it isn't. Because what this is, you can think of that as being a linear operator that depends on U star, that acts on U tilde. I will in a moment write down what L is, I'll write it down there. The linear operator is an object that takes a function v and it returns minus v plus the integral of w in y f prime at U star of y times v of y in dy. I have some feedback on this. One thumb up and the others. Would you be able to solve this equation if I gave you a formula for it? So the game we have to play now is to find, to solve this equation. So suppose I give you f, I'm giving you f prime, I give you w, and you're left with this equation to solve. Well, you can do several things. You can numerically try to solve it, and that's for tomorrow onwards. But I want to show you that if you make some simplifying assumption, namely you take, you put yourself in a region where there is literally a Turing bifurcation, you make your cortex being infinitely large, you make your kernel being distance dependent, so you make it homogeneous, and you make U star to be homogeneous in space, so not dependent on time, this becomes really a manageable equation to solve. So let's try to see what happens there. So now what I will be doing is I'm going to be switching notations here just to introduce what happens in a Turing bifurcation. In a Turing bifurcation, you consider an infinitely large medium. There. And let me just say this. I thought I could retain it on the board, but I can't. And you can revisit this, by the way, in the tutorial. you take your steady state, u star of x, to be constant in space. So you're putting yourself in a position that when you're changing parameter, your steady state can change, but it will always be flat as it changes. And you typically take a cortex, a W, a synaptic kernel that depends only on the distance between x and y. It's not really the distance between depends on the coordinates x minus y. What are typical examples of that? Well, one example that was popular at some point in neural fields is to say, I'm going to take the function capital W to be the exponential at minus absolute value of z. So I have locally an excitation, and then this excitation plateaus down. But the important thing is that I'm using only one coordinate to specify W, and then my kernel, now it's specified with distances, effect, x minus y. Another very popular choice is a kernel that is of excitatory inhibitory type. So you take kernels that are also distance dependent, but they don't have always a positive sign. So sometimes you would take W of z to be a constant, a1 e to the minus z in z1 squared, z squared over sigma 1 minus a2, two Gaussians, let's say, with two parameters a1, a2, and sigma 1 and sigma 2, so that you can get also some form of inhibition where you don't excite your neighbor, but you depress your neighbor's activity. Both of them are valid. And actually, there are also experimental evidence for that, depending on where you are on your cortex. So what happens under these assumptions? Well, what happens is that now the linearized problem looks like if you redo the calculations that I mentioned earlier on, now you have some simplifications. You have dt u tilde, dt u tilde xt is minus u tilde xt plus a convolution. You have, first off, there was an f prime on u star. But f prime on u star is a number that you can take out of the integral because it's not dependent on space anymore. And then you're left with just a convolution between the function big W and the thing you want to solve for. This is now a notable simplification because the moment I introduce a convolution, I have some hope to turn this into a problem that I can solve in Fourier space. So now, depending on whether this is a subject that you like or you hate, you will like or hate the next bit of this explanation. But the idea is effectively that you seek for solutions to this problem as inverse Fourier transform. Let me just write it down first so that I don't make mistakes. Inverse, so I'm integrating the big side. So you now postulate the fact that your pattern can be Fourier transformed in space. The forward Fourier transform is the function u hat. So let me think. Now, between hats and tildes, this is going to be a nightmare. This is a tilde. This one is a hat, and the u hat xit will be the Fourier transform of u x tilde xt e to the minus ixi x in dx. And so you seek for solutions of this form in Fourier space in which u tilde is the Fourier transform of your signal. So the way you typically progress at this stage is you take formally a Fourier transform of your right-hand side of the equation that you're trying to solve. And what are you going to get? If you now apply Fourier transform formally to the entire right-hand side there, you're going to get dt u hat psi t equals. So this one is minus u hat psi t. And then you take, there is a number f prime at u star. And then you now take advantage of the fact that this is a convolution. And Fourier transforms of convolutions in Fourier space are products. So in Fourier space, this looks like w hat psi times u tilde psi. So the big advantage of doing this business is that in here, in space, what happens at position x is coupled to what happens everywhere else in the cortex, because I have to sum over y. But in Fourier space, your psi appears as a decoupled parameter here, right? So now I can solve for each psi separately, like in slices. And this to me, now once you look at it, this problem looks like I have, what is the red? What is the red here? You have lambda psi times u hat psi t, in which lambda, this is an important parameter, equals -1 plus f prime at u star times w hat psi. I mean, this may not look like progress, but we're basically done. Because what we found out from the top equation, you can immediately write down, from this equation, you can immediately write down u hat psi of t is an exponential, e to the lambda psi times t times u hat psi 0. That's what happened when I solved this equation there. As I said, it's pointwise, so it's basically a linear equation. And then I can now plug back in here to find the solution in general. So I'm gonna have u tilde xt equals 1 over 2 pi. And over there, what I do is I convolve u hat psi 0 times e to the lambda psi t times times e to the I psi x in the psi. Yes. Now, I have the feeling I've been talking to myself for 10 minutes. So let's say, irrespective of what you get out now, I need to get some feedback for doing more of this or less of this in the board in the next 2 days. Okay, so not now, but perhaps at some point, drop me some feedback on that. Let's say if I've lost you, you can now reconvene here. What is happening is we are now, our objective is to establish whether u tilde will go to 0 or not as t goes to infinity. And we have a formula that tells us precisely that this will happen depending on lambda psi. Over here, I'm summing over all the psi's. And depending on the sign of lambda, if for a particular psi I have a lambda negative, my things are going to be dampened. If for a particular psi lambda is positive, that mode will be amplified. And linear stability, therefore, I have a sufficient condition for linear stability, that is lambda psi has to be less than 1 for all psi. So if my perturbations are to be stable, this lambda psi has to be less than 1 everywhere. Sorry, less than 0, what am I saying? Less than 0 everywhere, so that all the perturbations will be, all the psi mode will be damped, and I'm going to go back to equilibrium. what's happening across a Turing bifurcation is that you're changing P. And when you change P, you are changing U star and you are changing F prime. Because in the example I've made somewhere up there, the P was coming into F. So as P changes, U star is changing a little bit, so this number will change a little bit, but also it will change because the parameter enters F. And at some point, you pass from a condition where this holds to when this does not hold. Because you have a particular value psi in which this is violated. And this is then when you see a lambda psi positive, which is going to destroy the pattern that you're going to get. Just to give you a hint of what is generally lambda of psi. Well, lambda of psi is -1 plus W hat of psi. W hat is the Fourier transform of the kernel. So once I choose a modeling kernel, I have chosen a shape W hat of psi. And once you plot these shapes, they invariably, well, first off, they invariably, in the variable psi, they have to be even, because this is W hat as a function of psi, because, well, I've chosen W to be real value, so this is what I would expect here. Typically, and this is only characteristic, of course, this is now a sketch that depends on the particular W that you pick, but the W that one typically picks, they have a maximum. And so whether you satisfy this equation or not boils down to drawing an horizontal line at 1 over F prime of U star and deciding whether this line is above the maximum or it intersects the maximum. At the critical point, this is where you have found exactly this point here. So you have the opportunity to go through, well, first of all, let me just make a plugin for today's tutorial. In today's tutorial, I've set up the tutorial in such a way, if you are a theoretically minded person and what gives you the kick is just understanding this, you can do that with pen and paper in the first 4 questions of the tutorial. If instead you are more of the type, yeah, I want to see this stuff happening on a real in silico cortex, I let you build up codes that let you visualize what happens with this pattern formation. And perhaps if you want to form a group, you can do that also in pairs. So you may get more or less of that depending on what you like. But I have to be, I think it would still be helpful if you want to ask questions about this mechanism, because I don't anticipate that this would be trivial. Any questions on what we've done so far, what's written on the board? I'm condensing quite a lot of information in 20 minutes talk. Yeah? No? Yes.
Didn't quite get some of the steps in between, like from where you had the linearized problem, and then the line below. What are you
From
doing?
here to there?
Yeah, what are you doing?
So let's say that I could. So let me just do rewind entirely. Let's suppose that I have not written anything on the board. So how is this? How could I are you familiar with Fourier transforms back and forth? Even mildly. No. So Fourier transforms are mathematical tool that lets you transform a solution, a function of space, into a function in sort of some frequency domain. And they have useful properties. First of all, you can go from physical world, so physical space, to what's called Fourier space, frequency space, back and forth very cheaply. And this is how I'm doing it. So in here, if you give me a function in, let's say here, if you give me a function in physical space, physical means X, so real space, I can get a function in the Fourier space with the psi with an integral. Yeah, and the interesting thing is that there is these exponentials there. But you can also go the other way around. If somebody gives me a formula in Fourier space, I can do something. In fact, these look remarkably similar. There is only a plus and minus difference here, except that it's a constant. It's very important. And now I can travel from Fourier space back to physical space. So why are these typically useful gadgets? Is because as often it happens in mathematics, you can sometimes gain from transplanting the problem in a new space where things are easy, solve your problem in this easy space, and then take the lift back to see what the solution is in real space. Real meaning X. So what has happened, if you just disregard what's written on the board, is I needed to solve these equations. First off, I am just regurgitating. This is not my research. It's been around for 100 years. You just have to solve this equation, which looks horrible with an integral. But once I look at it with the lenses of Fourier transform, in that space, which is now written here in red, when I've got the psi, not the x, it looks very simple. I just got a product. So what I did there is I said, okay, let me take Fourier transform of everything. And this Fourier transform takes the derivatives into derivatives with xi. This one into this one with xi. And the simplifying thing is that the integral in physical space, in Fourier space, becomes a product. So that's very easy now to solve. And I solved it in an equation that I called tilde, that I could write down. And then I took the lift back, wrote it like that. And then I said, oh, what does this thing tell me? It tells me that I can blow up or not, depending on lambda of psi. And this lambda of psi I can study. And then I say, as soon as psi hits, as soon as lambda hits certain conditions, I blow up. Sorry, this might be not perhaps very clear, but I'm very happy to go through it again in the tutorial. Yes.
I have one question, it's like the P there, we put it in front. I also the convolution, how to
Which, I mean, I put a P here,
yeah,
which I then forget to write, and then I never replicate it again. This is just a parameter that when I change it, there are changing conditions in my system. For instance, when P changes, this is like, think of it as being a number 10, literally. I find U star, the equilibrium when P is 10, and now I'm saying at T equals 11, I change equilibrium. The reason why I could forgivably not include P is that the only place where P will enter is here, in this description. So then I said when P changes, U star changes, but also this number changes as a whole, because if I now put comma P, if I reinstate P, it will appear only here. And so as you're moving on this diagram, P changes and I find all lambda be negative. P changes, I find all lambda be negative. And at some point, oh, I now find that for one psi, I've got a negative lambda and a positive lambda just because I switch a critical point. Because my parameters, what is it? Here. My parameter changes and therefore it changes 1 over f prime. Here. And this means moving on this diagram. Moving on this diagram, the moment I hit this threshold, it means that somewhere, let's say in this case, I've got that for this value of psi, lambda is bigger than 0. And so what has now happened under the hood is that I have no longer a stable pattern, because in that band, all the psi's for which lambda is bigger than 0 are going to just expand in time. And that's how the instability occurs. Yes.
Or help me understand the restriction, or like what it means to have the equilibrium be a constant. This is like your entire cortex is firing at some constant rate.
Yes. All your, say, synaptic variables are homogeneous, let's say.
And is that only interesting because that's kind of like the base state?
Exactly. So this is really interesting because, so this is, Our mathematicians explain, so in fact, in all the experiments I showed you, you never saw flat. There were always some bits of the cortex that were just having higher rates than others. And so this is how mathematicians explain how a system, even in the absence of a forcing, because this can also happen when I of x is 0, how a system can generate this spontaneously. So the simplest setup is to say I have a completely flat cortex, which means absence of pattern. The cortex doesn't have to be dead, it's just at rest. Or it doesn't even have to be at rest, it just doesn't have to have differences in position. Yeah? And there is nothing about this cortex that makes it special. So there is not like I'm injecting current somewhere. It's just a completely flat state. And then upon changing parameters, because of certain conditions, I see that patterns generate, because that state can no longer be supported. I mean, it exists, but it's not stable. So any imperfection will take you away from it. And so naturally you see patterns.
So you're actually interested in when
The transition occurs.
okay. But you don't want these lambdas to be less than 0 if you want something interesting.
Exactly. You have to just switch to see stuff happening.
But if they're exactly 0, you have something stable.
If you're exactly 0, you're now interested in just when you have this condition there, you are in a situation that is called in nonlinear dynamics neutral stability. So you're neither nor, and it's typically you have to investigate what happens there. But the transition is more what one is interested in. Of course you can, yeah, of course. Let me just state, well, as we're winding down before lunch, a little bit. In fact, I'll probably stick with you. I'll take back the 5 minutes I gave you before. That this is not new. Not only is not new because it's not my research, it's research that's been around, but also the idea, it's nothing to do with neuroscience. So the idea that you've heard me saying, you've heard me calling Turing-like bifurcations, these things, and perhaps my only contribution to the field is to trying to call them Turing-like and not Turing bifurcations. So this is how any nonlinear reaction diffusion problem typically generates patterns of this type. So if you go back and look at how Turing envisioned this type of phenomenon, they looked at how two populations of reactants talk to each other, let's say, or interact with each other, and those are reaction diffusion problems. So those are problems that first off look mathematically a world away from these ones. They're models that have, they're PDEs, they're not integral differential equations. You have to have diffusion for a Turing bifurcation to happen. There is obviously no diffusion happening here. You need to have two populations to have any Turing phenomenon happen. So to have a Turing bifurcation, you would need to have at least two reactants. In here, there is only a single field that we're looking at. It's just a scalar equation. But believe me, if you saw what Turing did, you'd end up drawing exactly the same diagrams. You'd end up having the exact same conditions. These lambda of size called a dispersion relation, PDE, and even integral differential equations. But here we have no diffusion. There is only one variable that causes this, and that's why I call it a Turing-like bifurcation. There is a diffusion-less bifurcation of this type. Yeah.
If W is a specific kernel, can you get diffusion?
I wish I could tell you yes or no. Some people, when I say this is not a diffusive system, they will start cooking up W's to just prove me wrong. So it sounds like you may be one. I mean, I have no problems with that. But let's say, of course, you can decide. You can take W's that are so pathological that effectively you are, well, not pathological. They're so specific that you can really end up having a diffusion-like process. But I think where I'm coming from is that the moment you write down these operator, really the structure of everything I say really changes dramatically. So even if you gave me such kernel, I would probably still write it as an integral and just now I said I would try to, so there's something I want to tell you before just going for lunch, and it is that now and perhaps later during the tutorial, you will experiment with this mechanism for pattern formation. But I want to give you literally a 30-second, a 3 minutes glimpse at what happens if you stop believing these models. If instead of taking firing rate models, and this will be the only spot that I have in the entire 3 days in which I'm going to talk about non-rate models, if I'm going to remove, if I stop believing rate models and I start looking at single, spiking models, what does it happen to this mechanism? So literally, I'm now saying I know how I can form patterns with the neural field, but these patterns are seen also in other models. How can I do that? So I had planned to give you let me just not do that because it will have. Just gonna plan and give you an idea of how you can form patterns in models that don't have any rate in it. So these are models in which, what do these models do? Oh, I shouldn't do that. I should share right now.
Yeah.
Yeah, I don't want to touch my screen now.
But I also can zoom in.
No, it's all right. I think I'll do it just there. I want to give you an example of what these models do, basically. So this is now a pattern that is also bump-like the one you've seen in the Drosophila, but you will see that there is some form of pepper, salt and pepper pattern happening here, and there is nothing happening there. This is space horizontal and T is vertical. You can think of this being a ring as well. And what happens here is really activity, but through a model that has nothing to do with neural fields. And I don't have any time to go through it. I'll just tell you that these are one of those models that don't try to describe a spike with its own voltage going up and down. So just pictorially, think that this is now the voltage in the ith neuronal network. So I have a network of n neurons, finitely many neurons, let's say. If this was an Hodgkin-active neuron, what I will do as I ramp up here is that I would shoot up very quickly. My channels would start kicking in, and then I would go down back and get a spike with an actual form. In these type of models, instead, what they do is they say, I have reached the threshold 1. What I'll do is I'll stop the simulation. I will record a firing time here, and I'm gonna cast an instantaneous current to all my neighbors in the network. So that is this horrible signal here. That's basically a synapse, as you've seen it before. This is now a resetting potential. So what I do is now is I arrive here, instead of computing the spike, I stop, I emit a current that gets sent everywhere else in the network, and I'm getting reset to a 0 value there. These are called spiking networks, and the computational neuroscience community, it's much more into this type of model. So they make compromises of some type at neuronal level, but they're good gadgets to study neuronal network. And so let me just skip everything. I don't know, I would have thought that I would just cover this material. Let me just say one thing. What you're seeing there is, remember in the synaptic variable that we saw at some point, I told you that there was like this discontinuity and there was a rate of change at which the synapse would go down. That's the parameter beta that I'm putting there. And what you see is that this object that is now a bump of activity exists if beta is sufficiently large. Upon increasing beta means making a a faster synapse and also making it closer and closer to a Dirac delta type synapse, setting beta large. The bump starts to meander, starts to wander left to right. And as beta increases even more, you're gonna get waves of this time. So as beta increases, I start with a bumpy object that eventually turns into this wave pattern there. So why am I even trying to show you this here, except pleasing my own ego, is because from a mathematical standpoint, what happens, rerunning the exercise that we have done in this type of models is basically a completely open territory. It's really complete. Every of the elements that I have here is really, really weird. What does it mean to have an equilibrium now? In the path description, this pattern was a steady state pattern, so it would satisfy an equilibrium of that type. Remember, I have to set to 0 the left-hand side there. But in here, even in states where I am like this state, this is not a steady state, because for me to observe it, I have to go through neuron spiking at every time, yeah? So this object seems to not change in time, but actually it is, because microscopically, if you just select a neuron, you will see it going up and down, spike and down, spike and down. So that object is not even a steady state. Let alone that I have no tools available to linearize. I have no ways to just try and discern what happens in this Turing bifurcation. So what I would have told you had I had the time, is that this is actually possible in a paper that we have just come up with, and there is a lot of beautiful and painful maths to do. And what basically, it turns out that none of what I just told you holds true. So what we can prove in these networks is that the flat state, let me just perhaps draw a picture to close and then go to lunch. The flat state, so imagine this is now parameter. In here, before I was drawing something like that then becomes unstable and destabilizes. What we prove there is that the flat state exists forever and it's always stable. So it means that we were particularly intrigued by that, because for us, if the steady state is always stable and I don't destabilize it, how on earth am I forming the patterns? Turns out that the steady state is always stable, but nearby, there exists like an enormous, countably many waves. And these countably many waves are very, 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 very slow, to the extent that if you're hovering next to it, you get attracted to very slow waves. And very slow waves is what you see here. So what happens here is that even if the pattern seems that you're getting a stationary state, you aren't. You're just being pinballed like in a chaos field, in which each site that you visit is a wave that is almost stationary. And once you increase beta, you start seeing movements of the pattern, because your waves become more unstable and they slot you stronger every time that you visit them. So this is just perhaps the take-home message, is that there is a lot of beautiful mathematical and physics research and computational research to be done if you choose different models that we're not going to discuss for the remainder of the course. But the mechanisms for the formation of patterns change completely. In this case, you don't even destabilize anything. These objects just exist, they're there. They just form a canvas for your dynamics to be walked around. There's no destabilization going on. I think I'll call it lunch if we all agree.
Yeah.
And yeah, see you later.
Yes.